Now, in the last video, I talked about allopatric versus sympatric speciation as two opposite ends. All right, so the allopatric was when you had differentiation with a barrier, and sympatric is when you had it without it. But in actuality, it's a little more complicated than that, and actually, speciation happens in a spectrum of different kinds of speciation, and it's actually sometimes a combination of several of these, which actually ultimately leads to speciation between the different species. But we're going to talk about in this video about several kinds of ways in which organisms separate, and we'll talk about it intellectually, and then I'll refer back to the examples that I gave in the previous videos so that you can tie it back to that and see the different kinds of speciation that exist. So this is all because some scientists distinguish between the kinds of speciation, which in the previous video I put as sympatric speciation. By the way, the most easy to understand is allopatric speciation, because you separate the species, they evolve separately in separate environments, and then you lead to differentiation evolution. It's a little harder to understand how species evolve within the same ecosystem or on a close ecosystem, where the gene flow can still exist between the two different branches which are splitting. But what I need to understand is that there's a lot of different ways in which that kind of same ecosystem speciation can occur. And so in other words, it's trying to get the sympatric speciation we discussed in the last video and splitting into a continuum. And that's what we're going to talk about in this video. So this could happen in a lot of different ways. The first one is that we talked about already is the idea of allopatry. Now allopatry, of course, allo means different. So this is when each species will develop within its own range, in other words, within its own kind of environment, and because the environments are different, genetic drift or the natural selection, coupled with mutations that occur individually in each environment, will cause local adaptation to that environment, and that's going to make the species change. And that's the easiest one to understand. Then you also have sympatry. And that's when many varieties will develop in one range, all right? Because the species becomes adapted to different aspects of the range. So in other words, that encompasses the whole concept that we talked about, the many different ways in which a species can actually develop all within the same ecosystem by becoming adapted to a special part of that ecosystem. But you could break this down further into it's kind of like subtypes. The way I think of sympatry is basically genetic isolation occurs without physical isolation, while in allopatry, the genetic isolation happens because of physical isolation. Okay? So there are several ways in which sympatry can happen, though, and that's the key that makes scientists try to differentiate between them. But if the genetic isolation happens without physical isolation, we call it sympatry. Okay? Now, sometimes there is somewhat of an isolation, so it's not really sympatry or allopatry, it just got somewhat of an isolation. Here's an example. So the variety differs at the edges of the range. So in other words, there's almost separation between them. So that means it's not truly separated, but it's almost separated. And that's going to include peripatric and parapatric. Now, peri means close, and para means far. Now, that means that peripatric will occur similar to allopatry, and then parapatric will happen similar to sympatry. So the idea here is that in peripatric, you're going to be almost physically isolated, and that's going to lead to genetic isolation. So it's nearly allopatric. In parapatric, you're going to be almost genetically isolated, and that's going to lead to physical isolation. So it's nearly sympatric. So in either case, is almost separating it that leads to actual speciation. I mean, gene flow is still going to be possible back and forth here, but it's going to be separated most of the time. So to give examples that we talked about on this, an example of parapatric would be, for example, when those sexual selection with the cyclids, right? Because the cyclids took different jobs within the environment because there were mutations that occur in their coloration pattern, which led to differential sexual selection. They chose to mate with people who looked like them, and that led to differentiation, sexual selection, and then ultimately to speciation. So that would be parapatric. Basically, the genetic difference, which almost isolates between them, ends up actually isolating them and creating speciation. So it's nearly sympatric because the gene change came first. Now, if the physical isolation came first, then it's nearly allopatric. So an example of that would be, for example, the maggot flies, which kind of separated themselves from each other because they moved into an adjacent niche. They kind of had an isolated niche because some live in some of the apples and some live in other apples, even though they were within an ecosystem that was close to each other. Because the niche was separate, they actually led to separation between them. So there was isolation first, and that led to isolation in the species. But remember, they technically could have had gene flow between them because there was not a true isolation. But it is also not really pure sympatry because they aren't completely integrated into the same range, you know. So they're kind of somewhat isolated. So I hope you understand what that means. And I'm going to do a review of all of this at the end, so maybe you can understand it. Now, this one is when it gets a little more confusing. This is called stasipatric. Now, this is when a new species formed because of chromosomal changes within the same range, but without adaptation to local differences. So, an example of that is the flower thing that we talked about. When autopolyploidy will take place, and then you're going to make a polyploidy flower, like the strawberry, for example, that became 4N. That strawberry can live in exactly the same ecosystem as the original. It will do exactly the same job or that the original strawberry did, but that happened within the same ecosystem of the original strawberry. It's just kind of like a chromosomal change which led to the difference between the species. But it's basically doing the same job. It looks almost the same, and so forth. So they're obviously compete with each other for that niche because they're trying to assume the very same niche. So that's why it's called stasipatric, because they are trying to occupy the same niche even as they change, and that's obviously going to increase competition, and ultimately only one of those species will probably survive. So this is usually because of a number of chromosome changes. By the way, chromosomal changes have been observed in animals as well, leading to speciation events. It's not just in plants. Things like that will happen in animals as well. Another weird kind of speciation they can have on the spectrum is called dichopatric. And dichopatric is the idea of being, you know, like dual citizenship. It's kind of like you belong to two different ranges. So that means one range will have so much variety within that range that the species can become different through adaptation to different aspects of the range. It sounds a lot like sympatric to me. So it's kind of hard to understand the difference between this one and sympatric, or this one and parapatric or peripatric. But the idea here is that there is no separation between the ecosystems. There is no new niche. It's more like, you know, one species got higher in the tree, some species went lower in the tree, another species went lower in the tree, and then like the warblers example that I talked about. And so that will be dichopatric because there's two different regions within the same ecosystem, and each one adapts to one region of the ecosystem just because of pressure to establish a different niche. And so you kind of create a niche to actually avoid the other group, and that's what leads to speciation. So there isn't a separate niche. You just make up a separate niche because you don't want to be in competition with the other group. That's kind of how I like to think about it. So let's review them all and kind of put it all together in a spectrum that you can actually understand. Allopatric is genetic isolation because of physical isolation. So the physical isolation is what leads to genetic isolation. And the allo means different, so that's a new way to remember that. Peripatric means close, and that's when it's similar to allopatric, but then you're going to have almost physically isolated, and that's going to lead to genetic isolation. And so it's nearly allopatric, and that's because you're going to assume an isolated niche. An example of allopatric, we did several. We talked about, for example, the squirrels in the Grand Canyon. We also talked about the salamanders in California. So those were allopatric. In peripatric isolation, the example that I gave you there was the maggot flies that actually separated to different niches within the same kind of range, so that it was an adjacent range kind of. They still could do gene flow between the two of them, but because they are assuming an isolated niche, they were actually separating from each other. Then I also talk about parapatric, and para means parallel, so you are within the same range. You can still kind of switch back and forth, and it's kind of like an adjacent range or at the edge of the range, and that's because of an adjacent niche. So it's almost like sympatric isolation in the sense that the change in genes happens first, and that will lead to the isolation of the niche. So the difference is that from peripatric to parapatric is the order. On peripatric, you change genetically because you chose a different niche. On parapatric, you choose a different niche because you changed genetically. So it's a little more sympatric. An example of that would be the cyclid with the colors. They had different colors and then they pair themselves up with people who looked at colors. So the genetic change preceded the isolation that led to their speciation event. So that's parapatric. Then you have sympatric, and that's when within the same population, because of genetic isolation, in other words, gene changes occur without exploring a different niche, and that leads to differentiation in population. All right. Then of course you have the stasipatric, and that will occur because of chromosomal changes. You can call it a special type of sympatry, but that actually sometimes leads to differentiation between the species, which tries to assume exactly the same niche. And you have dichopatric, which is when a separate niche develops, all right, because without isolation, because of pressure to assume a different niche within the same range. So you create a niche. That's not that the niche is available, because there's pressure for that. So these are the different kinds of speciation that occur in biology. And as long as you know that there's a difference between allopatric and the other kinds, you probably should be okay. But remember that sympatric speciation sometimes encompasses all the other kinds which are not allopatric kinds. In other words, any other time that you're not going to have actual physical separation, a lot of people kind of group them all together into a speciation without physical barriers. So there's the major category of dividing, which we talked about in the previous videos, which is barrier versus no barrier. But then you can also look at other kinds of ways to separate the barrier. In peripatric, it's you separate because of exploring a different niche that's available. On parapatric, you change the genes and then you explore a different niche. On sympatric, you explore the same niche, but within the same ecosystem, but because of a genetic change that occurs. And then on dichopatric, you develop new niches because of pressure to do so, and that leads to isolation, that leads to genetic changes. And on stasipatric, you change the chromosomes of the species to create new species between them. And remember, of course, the example of stasipatric was the flower thing that we did, and the specials of dichopatric was the warbler example that we did. All right, I hope that this makes it all clear and you understand a lot. And on the next video, we'll talk about evidence for macroevolution, so that you can be like, does all of this even happen? Is there any evidence for this? We'll see you then.